Hello, and thank you for being here today. Let's talk about potential changes for Darwin Core and the Global Biodiversity Information Facility, or GBIF, publishing model, and their relationship to collection management systems. I feel like I am standing at a crossroads summoning a demon, but here goes. First, let's touch on GBIF's unified model, which was or will be discussed in this symposium. The new model recognizes that biodiversity data are more complex than just the occurrence of species in time and space, and is looking toward the aggregation of data such as automated monitoring, biotic interactions, and environmental measurements. In addition, the model expects to address an issue collections have known about for a long time, that we need improved handling of data representing material. The new GBIF model is being tested and matured to accommodate real data sets, and I was able to participate in this as part of the Arctos data test case. In conjunction with GBIF's planning, it just so happens that TDWG convened a new task group last year to address physical objects. I am the convener of the task group. We are making progress toward a proposal for a clarification of the Darwin Core material sample class with its own properties, and expect the outcome of this process to be a standard for sharing more complete information about biological specimens, including their physical properties and associations with each other, organisms, and research products. As we began our work, there was quite a bit of philosophizing, but we settled on the need to first achieve a clear conceptual delineation between the terms material sample, preserved specimen, living specimen, and fossil specimen. We determined that preserved, living, and fossil specimen are all just types of material sample, and that we likely don't need 4 classes of the same general thing. So we have focused on material sample as the most broad term of the 4. We have worked to give that term a better definition and on a recommendation for the other 3 terms, recognizing that they will need to be considered as we develop material sample type. This is still a work in progress, and we are trying to be mindful of the potential opportunities the new GBIF model may afford, and the fact that many will choose to continue publishing under the current model. The Darwin Core material sample term and GBIF's material entity fit well. Given this, the Material Sample Task Group realized that identifiers will be crucial in the new GBIF data model, and that material will benefit from good ones. We are also mindful of the fact that as the occurrence class has been expanded over time to include material type descriptions, some terms may need review and may need to be organized as properties of material sample. Finally, we recognize that some new terms may be needed. The ability of GBIF to handle richer material data and a beefed up material sample class in Darwin Core will combine to create both challenges and opportunities for collections managing material data. Let's look at some challenges. Challenge number 1. Stop thinking about objects as occurrences and start thinking about objects as participating in occurrence events. Museums have been forcing their object information into occurrences to meet the needs of GBIF, when the occurrence or collection event is really just the first event in the life of a museum object. We are now conditioned to think of the data about our objects as occurrence data, and changing this thought process will be challenging. Not to mention the challenge of mapping data that has sometimes been formatted specifically to meet the GBIF occurrence standard to something new and far more descriptive. Challenge number 2, what's a material sample? We don't generally accession individuals or objects that we don't plan to keep forever. The image on the left is of a mouse before or perhaps as it was being collected. Is this a material sample or an organism that results in a material sample? In this case, the object cataloged is a whole organism preserved in ethanol with the catalog number MVZ-MAM23964. So it's kind of both? But things commonly come and go. The image on the right is a raccoon after its collection and preparation for storage in a museum collection. In this case, the objects cataloged include a study skin, skeleton, and frozen samples of heart, liver, kidney, and muscle. All of these parts share the catalog number DMNS-MAM16640. Is the material sample the original whole organism or the preserved parts created later? After a lot of discussion, the Tadwig Material Sample Task Group decided that an organism may be a material sample, but a material sample does not necessarily need to be an organism, which means the answer is it depends. But this also ties into challenge number 3. Material samples are better with good identifiers. In most museum collection catalogs, there is a mix of single things with a catalog number and multiple things that share a number. As the data gets extended and the various objects sharing a number get used in different ways, it starts to become clear that each thing needs an identifier, and not just any identifier. If we are going to realize the extended digital specimen and make the best use of GBIF's new data model, objects will benefit from globally unique and persistent identifiers. Minting these forever identifiers at every stage is burdensome and probably just adds confusion. So the challenge is, when do we assign these and how will they be maintained? But remember, we will also need good identifiers for the collections these objects are housed in, the events these objects participate in, the people who interact with them, the taxa they represent, and, well, the bottom line is that everything will need good identifiers, and this is very much a broader community challenge. Which brings me to challenge number 4, acceptance and use. Extending specimen data and aggregating under GBIF's new data model will likely require some social engineering. Collection managers will need to get used to working with new kinds of identifiers, potentially minting them and instructing others to use them properly. This means that curators and people who use materials will need to support something other than the traditional catalog numbers being used in things like publications and even in the collection itself. It also means that publishers will need to support and potentially require non-traditional identifiers if we ever hope to trace research results back to the objects that created them. This is an even broader community challenge, requiring the participation of everyone involved in the chain of information related to these objects. But enough about the challenges, let's look at some opportunities. The biggest opportunity for physical collections provided by GBIF's new model and an enhanced group of properties for Darwin Core Material Sample Class lies in the ability to showcase and track the objects we care for in an aggregation. It also lets them showcase all of the details and information that we know about these objects in a way that lets researchers filter and select the objects appropriate for whatever it is they are doing. The second opportunity afforded by the new GBIF model is the ability to tell your own story. With the current GBIF Darwin Core archive, which is flat and focused, there is one entry point and one story to tell. Given normalized data, anyone can hop in from anywhere and tell their own story. For example, you can build yourself a publication-centric picture and treat the dead rats like secondary metadata. Opportunity number 3 is like magic. It involves connectivity, most of which gets lost in the occurrence model, but can be allowed to shine in a post-occurrence approach. Links to information will appear in the new data model. Someone publishes a bunch of genetic sequence data that uses your material sample identifiers. GBIF can match these and yay everyone! Anyone can now follow the chain of information from specimen collection to genetic sequence and even from collector to researcher. This is where IDs that are or can be made into URLs of some form can do stuff. A bit more investment results in a lot more functionality, like this Crossref example, which exists because DOIs can be magicked into URLs. So now we return to challenge number 4, which for me is also an opportunity. Connecting our data in a rich mesh of knowledge will require community support and participation. Support for collection managers and the systems they use to maintain the knowledge, and support for all of the players who help make connecting data possible. We all need to work together to create systems and processes that work for everyone. We will all have to make compromises in a connected and vibrant community of biodiversity data providers, publishers, users, and standard-setting bodies. But in the end, that will result in better outcomes for everyone. So, from the crossroads, I say thank you to Dusty McDonald, the Arctos programmer, for his enthusiasm, John Vichorik for his experience, Tim Robertson for his patience, the Tadwig Material Sample Task Group for their time and attention, the Arctos community for their commitment to collaboration, and thank you for listening.